And without further ado, it's a pleasure to welcome Will Valdar from UNC Chapel Hill for our very first talk. Thanks very much, Will.
Thank you very much, Reinmar, for organizing this conference. Good afternoon, everybody in Europe. Good morning, everyone in the US. So this is the title of my talk. What I'm actually going to talk about in order is I'm going to say a bit about X-chromosome inactivation and describe X-inactivation skewing and these things called XE alleles. Then I'm going to describe a collaborative cross-RIX experiment that studies X-inactivation skewing and finds a new XE allele, and then also finds something kind of hidden and I think it's pretty cool, which is it estimates the number of brain precursor cells in the day 5 mouse embryo. And this is work done primarily by my student, Kathie's son, and also with the help of all of these people. And this is part of a collaboration between myself, Lisa Tarantino, and Fernando Pardo-Manuel de Villena, henceforth Fernando. So as most of you will be aware, for most females, the cells of the for most species of mammals, females are XX and males are XY. And so naively, one might think, well, does that mean that female cells have twice as much expression from the X chromosome? And in fact, that's not true. What happens is a form of dosage compensation, whereby one copy of the X chromosome is inactivated. This is a complex, multi-layered epigenetic process. It basically becomes heavily sort of coated in heterochromatin, turns into a bar body. And this is termed X chromosome inactivation. So let's just look at a female cell and let's imagine that we've got one chromosome that's one X chromosome that's orange and one X chromosome that's black. And so the way this works is that at around day 5 in the developing embryo, there's this process that happens whereby every cell randomly decides whether it's going to keep active the black or the orange. And so I've indicated here, the orange balls are the cells that have decided to inactivate the black and keep active the orange, and vice versa. And this decision of which chromosome to inactivate, this is propagated through all of the daughter cells of each cell, such that when you get to the adult tissue, you get these clusters of descendants from those cells that carry on that cell fate. And the classic example of this is the calico cat. All calico cats are female, and they have this kind of blotchy, these blotches of orange because the orange cells in this case, they're expressing genes for orange fur, and they have this sort of orange and black pattern. So the classical understanding of X inactivation ratios, which is really what the proportion of inactivation is of one of the chromosomes versus the other, is that it's random and independent in each cell. And such that if we had a population of females and we for each one we looked at their cells and we looked at the proportion where they inactivate one chromosome versus the other, those proportions, those overall proportions, they would vary, but they would vary in a distribution that kind of looks like this, that's centered around 50-50. So in the mouse, there's evidence that this process is actually under genetic control. So okay, we've got the mouse chromosome here and highlighted there's the X-inactivation center, which has got various sort of genes and bits and pieces that orchestrate X-inactivation. But the important thing in mice is that people have found that there's this region which is not very well defined, but it's a region called the X-chromosome controlling element, or XCE, and it's got a number of different alleles. And so different inbred strains, for example, have different, they're homozygous for different alleles. And these alleles form an allelic series, which I've labeled here, going from weak to strong. What does that mean? Well, first of all, let's imagine that we've got an individual who is heterozygous at the X chromosome at the XE allele, and so if they're homozygous, then you just see this 50-50 ratio. Okay, just as you might expect. However, if they're heterozygous, then you see something different. You see skewing. So if we look at that purple line there, you can see this is for an individual where on one copy of the X, they've got XEB, and on the other copy of the X, they've got XEA. And there's this real shift towards XEEB. There's this preferential inactivation of XEEA in favor of XEEB, hence the weak and strong series. So that picture was actually from a study that was done between my lab and that of Fernando. And that was a while ago. The general way in which we estimated these X-inactivation ratios was as follows. We took 2 inbred strains, made an F1 hybrid, and then harvested tissue from whatever organs we were interested in. And then we calculated, so we got the gene expression, and we looked at transcripts for heterozygous X genes, and we calculated allele-specific expression. Fairly simply, you know, you do the you get the proportion from just the number of orange versus black. So fast forward a bit, and Fernando, Lisa Tarantino, and I were still working on an experiment that uses collaborative cross mice. This is a collaborative cross, 8 inbred strains that are used to generate these 75 odd recombinant inbred strains. And the experiment we were working on wasn't really anything to do with X chromosome inactivation, but it had this kind of structure. So this is really a series of F1 crosses between CC strains. And so one might ask, okay, well, what is there actually to discover, right? You've got this experiment. What about XE is there to discover? Well, some of the inbred strains, the founders of the collaborative cross, we know, or we have a very good idea of what the XE allele is. Two of them, however, we only know from phylogeny, and so we don't really know because there haven't been any experiments, and so these are predicted to be B, but you know, we don't know that, and so we thought, well, we could use this data set to answer that question. What we actually ended up using was 2 data sets comprising 28 CC strains combined to form 29 CCF1 lines, and a total of 266 female samples. So the way we estimated X-inactivation ratios was we did RNA-seq in brain, because that was the tissue we were interested in, 100 base pair single-end reads. We used a KEMA-based approach to get ASE, and we used a sort of fancy Bayesian hierarchical beta binomial model to estimate X-inactivation proportion per gene, per pup, per cross. And what the results of that look like is this is just a sample of the results here on the x-axis. We've got the estimated XEI proportion, and this is actually for a cross where at the XEE allele that it's homozygous, so we would expect it to be a 50-50 ratio. These are the X-inactivation proportions for the individual pups. This is the overall sort of consensus proportion for that F1 cross. When we look at this picture for an F1 where they're heterozygous for those XE alleles, we see this shift just as we would expect to. And so the sort of general results from here is that when we look at XEE alleles where we know what they are and we know what their strengths are, we see results that are consistent with what's previously been reported. And here, gray is consistent, purple is slightly inconsistent or somewhat inconsistent. But when it came to any crosses that involved NOD at the XE locus, we saw something very inconsistent. We saw that there was tremendous skewing. And what we learned from this was that NOD has a novel and unique functional allele XEG, which is the weakest in the allelic series. And we also looked at the sequence and found some interesting things there to do with likely sort of duplication events that might give rise to this. What I want to end on, though, is talking about something which I think is pretty cool, which is we've got this picture of the way that these mice proportions vary around the overall cross consensus. And yeah, this is a stochastic process. We would expect them to vary, but the way in which they varied was fairly consistent. And the extent to which they vary is interesting in itself, because it is actually telling us something. And to see what it's telling us, it's useful to think about this thought experiment called the Pólya urn. So the Pólya urn is this sort of imaginary urn, and it starts off with some number A orange balls and B black balls. And the way the process works is you randomly put your hand in and you take out one ball at random. And then you put back in two balls of the same color. Okay. And you keep doing this. You keep doing this process, and after an infinite number of draws, you have lots and lots of balls, but the proportion mu of orange balls looks like a particular shape, like the histogram of that, if you do it for lots of urns, looks like a beta distribution with parameters A and B. And so the general kind of intuition here is that if the number of balls you start with was small, then the proportion of orange balls at the end was going to vary a lot from urn to urn. But if the number of balls you start with is large, then the proportion is going to vary only a little. And if you think about it, that will make sense. So the metaphor here is obviously that the starting urn corresponds to the day 5 epiblast. The ending urn corresponds to the adult tissue. And the proportion of orange balls corresponds to XEI proportion, right? The X-inactivation ratio. And so the intuition here is that the variation in XEI proportion going from mouse to mouse tells you something about the number of starting balls. In our case, it tells us about the number of cells in our measured tissue, the brain, the number of brain precursor cells in that day 5 epiblast. And that number we estimated to be between 20 and 30 brain precursor cells. This is a posterior distribution that we backed out of our model, and the blue dotted line is showing the consensus. So that's kind of cool, because if you imagine a day 5 embryo, none of those cells know that they're going to be brain precursor cells. There's no indication that that's going to happen. So we're actually estimating something that at least in this decade, be very difficult to discover experimentally. And this is sort of hidden, interesting thing. That's all I'm going to say. Thank you to all of these people. If you want to read more about this, then you can look at our paper, which is down there. Thanks very much.
Thanks, Will. That's fantastic. Really fascinating talk. We've run a little bit out of time, but there's a number of questions in the chat, Will, if you maybe want to take a look at this later, but I think we got to move on to our next speaker.